Okay, chapter 4, review, is going to be a little bit about Pavlovian conditioning and some of the questions you had on the poll, and hopefully we can clarify everything before you take the probe. We're going to arrive at a conclusion that we often refer to as biological constraints on learning. I'm also going to talk briefly about discrimination and generalization, so we have that down. First of all, I want to say that your guys' comments on the wiki and what you're producing there is really awesome. The content has been great. I've been placing comments usually later in the evening about some of the things that I think are really cool, or if there are alternate interpretations or maybe problems, I might comment them on them as well. One of the things I noticed from the poll and from the wiki is I probably put too much emphasis on the Rescorla-Wagner model. The Rescorla-Wagner model is great stuff. It helps us predict a lot of phenomena in Pavlovian conditioning, but it's pretty advanced stuff. What I wanted you to recognize that it's a quantitative model of conditioning. It's been very influential. It's been it's somewhat controversial still. It's been studied very heavily. And it's pretty neat, but beyond that, the details of the math and so on and so forth, I think Mazer and I need to take it easy on that a little bit. I think it's, I had a whole class, believe it or not, in graduate school on the Rescorla-Wagner model. It can get really, really heavy. And for this class, I think we have bigger fish to fry. I think we got other phenomena and other principles that we need to go through. So, what I wanted to start with in this lecture is a brief overview of what are sometimes referred to as general principles of learning. This was an important question for experimental psychologists. Early on, they were trying to discover the universals, what we might say. They were trying to find these general rules that applied broadly, applied to people, applied to non-human animals, applied in a variety of situations and contexts and so on. The counter question of this is that is learning dependent somewhat on genetic makeup? And we'll see that, basically the answer is somewhere in between. It's kind of hard to come up with a universal for anything. All you have to do is find a single exception. If you say that all people are X, all you have to do is find a single exception to that to disprove that all people are X. So general principles of learning were difficult to support, and as you'll notice, and as you may have noticed already, the experiments that we talk about are experiments demonstrating how general principles do not hold, or what the exceptions are. However, the reasons for a lot of experiments was that people made pretty bold claims about general processes of learning. The three that were mainly put forth early on were the principle, or the principle of contiguity, the principle of frequency, and the principle of equipotentiality. Okay, so let's review these. These were proposed as universal laws, so to speak, or general processes of learning. And contiguity is that the CS and the US must be close in space and time. In order for effective conditioning to occur, the CS and the US must be next to one another. They must be happening. You'll already notice, or maybe you've noticed, this is a major hypothesis in Pavlov, and that is in trace conditioning. That's when the CS is off and the US comes on a little bit later. The word trace conditioning comes from the fact that Pavlov thought that there had to be a memory trace, like a reverberating circuit representing the CS, bouncing around in your brain, if you will, in order for effective conditioning to occur. What he needed was something close in space and time to the US in order for conditioning to occur. It was a requirement almost of his theory. That's why he called it trace conditioning. He couldn't explain how two stimuli could go together if there wasn't something mediating in between, a memory trace. That's, as we'll see, that's difficult to consider. So Garcia in 1966 does an experiment where he presents a CS, and then in delay conditioning, there's this was trace conditioning. The CS comes on, it goes off, and then there's a long delay, And then he gives the US, which is lithium chloride, which makes animals sick. He lets rats drink sweet water, takes the bottle away. 22 minutes later, he induces sickness with a lithium chloride injection. This induces vomiting, gut wrenching, and so on. Rats don't vomit, but they were visibly upset and sick, and so on and so forth. The next day, when he gives them sweet water, they avoid it. They completely avoid it. Now, it's not because these rats haven't had anything. These rats haven't had anything to drink for 24 hours, right? They're water deprived. Now, you might think, boy, they're thirsty. Matt, why don't you call the rats thirsty? And the reason I don't like to use the word thirst or thirsty is that I think it is an intervening variable. We drink for a lot of different reasons. We eat for a lot of reasons other than a hunger. And it's not clear what hunger and thirst are necessarily. I mean, physiologically, you can say, oh, well, hunger is this or thirst is that or whatever. But that winds up being not true all the time. And people drink water for a lot of reasons. People eat food for a lot of reasons, not always just when they're hungry. So to explain why someone eats as they're being hungry, it doesn't do it for me. You still have to explain the hunger. Now, if you say an animal drinks because it hasn't drank for 24 hours, I think that's a little bit better explanation. So these rats had not had water for 24 hours, yet they still avoided the sweet water. So, S. Gordon Stevens later found that the 24-hour delay was effective in this conditioning. You gave them sweet water. 24 hours later, you made them sick. The next day, they would avoid sweet water again. So this phenomena we call taste aversion learning destroys the contiguity principle. And we've all experienced this. Taste aversion learning is so robust and so well studied. We actually use it experimentally to show other things, to look at other things. But you may have even experienced it or had a friend experience it. I had a friend, we came home from college after our first year. We went out and had a good time. And Jeff ate a whole bag of nacho cheese Doritos and then got really sick. And never wanted to eat nacho cheese Doritos again. In fact, couldn't even stand the smell. If he saw them around, it would induce sickness in him. This is taste aversion learning. We know that pregnant, expecting mothers are more sensitive to taste aversions. There's all sorts of interesting phenomena around taste aversion learning. It completely destroys the contiguity principle. The CS and the US don't have to be close in space and time. They can be separated by hours and still be effective. We've actually found taste aversion learning useful in the laboratory to study various phenomena, but we've also used it to explain certain phenomena and used it as a therapeutic tool, so to speak, in a number of contexts. You watched the lion taste aversion attempts. There were attempts also to create a taste aversion to sheep in wolves in the west of the United States. But if you can induce a sickness to livestock, maybe natural predators will avoid those livestock. That's how it's been used. It's also been used for, or attempted to get animals to stop eating crops. The other thing is we now understand maybe that what's going on in chemotherapy, or one of the side effects of chemotherapy, is taste aversion. We have this phenomena of children who had cancer and were about to go into chemotherapy. And what do you do when you're ready to go into a really painful, horrible procedure? Is you have your favorite meal, or you have your favorite lunch, or your favorite dinner. And so these kids were having their favorite meal and then going into chemotherapy, and then they were done and cancer-free, never wanted to eat that meal, that food ever again. And people were wondering why this was happening, and it may be because of taste aversion learning, or that's the most likely explanation of this. So instead, now instead of producing these taste aversions toward kids' favorite food, what researchers have been doing is giving kids food before chemotherapy that's very unique in flavor, very unique in texture and color, and so on. And then if they get sick and don't want to eat that food ever again, oh well, they avoid one of the preferred foods to give kids going into chemotherapy is lime jello. It's very uniquely flavored, very uniquely textured, and so on. And if they avoid it, they avoid it. They if they're averse to it, oh well. If I don't eat another spoonful of lime jello, my life probably wouldn't be that. I wouldn't miss much, right? Okay. The second general principle of learning that's been proposed is the frequency. The principle of frequency, and that is for effective conditioning, the number of CS-US pairings is critical. So, in a condition suppression paradigm, we could run one group that gets 3 CS-US presentations and another group that gets 3 CS-US presentations. But the second group also gets pre-exposure to CS. Now, if frequency was the key here, there are 3 pairings in both groups. So these groups should equally condition, but it's not true. If we run the test, we find that this group, group 1, does much better. As it has, we see the condition response more robustly in group 1 than we do in group 2. This phenomena, we may not see any conditioning at all in this group. These are conditions, these are adequate conditions for what we call latent inhibition, and that is that if we have CS pre-exposures without the US, that the CS sort of becomes irrelevant. You don't pay attention to it, and then when you present the US, it doesn't really help. It doesn't really condition at all. Now, a lot of people might say, well, that experiment is not well designed, or there's some problems with it. But Rescorla did was a critical series of experiments in 1966 and 1968 in condition suppression. Again, he presented 3 conditioning trials, 3 tone shock pairings to 2 groups, and then asked the question which group shows better conditioning. And of course, group 1 showed robust conditioning, whereas group 2 didn't. And what you can see here is that there are CS presentations followed by nothing, and then there are US presentations that were preceded by nothing. This is sometimes called the random control group. We find no conditioning with situations or arrangements like this. So frequency is not what it's about. Equipotentiality was the third general principle of learning that was proposed, and the experiment that called this into question is so famous, we have a name for it. We call it the bright, noisy, tasty water experiment. And it's Garcia, 1966. And again, this is like a taste aversion learning experiment, except what we do is we have 2 groups of rats. One group receives what we call bright, noisy water, and that is, as it licks on a water bottle, a light flash and a click, sort of like a clicker, your mouse clicker sounds. Okay, a second group gets a water bottle and it has sweet water in it. It has saccharine water in it. This is called the bright, noisy water. This is called the tasty water. Now, we take group 1 and we divide them in half. Half the animals are made sick with lithium chloride. Half the animals are shocked following drinking bright, noisy water. Following tasty water, half the group is made sick with lithium chloride. Half the group is shocked. Then the next day, we offer them the water that they were drinking previously. So these 2 groups are then again offered bright, noisy water, and what do we see? We see that this group does not condition. If this is a condition suppression or a taste aversion, then what animals should do is, or this is some sort of aversion learning, the animals should avoid the bright, noisy water, but they don't. They drink normally. The animals that were shocked are the ones that don't drink. So conditioning, so the light flash and the click were ineffective when the US is lithium chloride, but it was effective when the US was shock. Okay, now you might say, well, shocking animals is going to lead to them not drinking. It doesn't matter what the water is, but that doesn't work out. The animals that are shocked after receiving tasty water drink normally. So when the CS is the taste, shock is an ineffective US, or the CS is not conditionable when shock is the US. Is shock effective as the U.S.? Yes, it is. It is over here. You could see that shock is effective as the U.S. So now the last question is lithium chloride effective as the U.S.? And that's what this last condition demonstrates, is that when the CS is sweetness, then lithium chloride is an effective US. So we have evidence that lithium chloride is an effective US, that shock is an ineffective US, but not all stimuli are equally conditionable. It depends on the nature of the US. So, contiguity is not necessary, but it actually turns out that it's fairly important. Frequency doesn't tell the whole story. It's important. That's still important. It doesn't mean, but it's not the complete answer. And not all stimuli are equally or effective or conditionable. That's a summary of those findings. So, you might ask, are there any rules? And I think there are some rules, or there are some general principles. Now, the first thing that we have to remember is that the US is some biologically important event. And I think the rule that we need to think about is, does the CS predict the US? Does it give the animal any information? Does it provide any expectation for what's coming? This helps explain this prediction sort of view of Pavlovian conditioning. This expectation view of Pavlovian conditioning can explain, for example, the effects of spaced versus mass trials. Remember last lecture, spaced training is more effective than mass training. And it turns out that maybe an explanation of this is that trials are so fast in mass training that you don't need prediction. That the U.S. is just coming and coming and coming, and it's coming rapidly, and it doesn't really matter. You don't have to. You just got to prepare. You're not going to condition the CS because the CS doesn't give you any good prediction. This might explain our frequency effects, and that is that if the CS occurs at other times without the U.S., or the U.S. occurs without the CS, even though they might be as frequently together as other situations, if the CS comes at times without the US and the US comes without the CS, then it's of no predictive value. It doesn't help you predict whether the US is coming or not. Blocking is also explained by this because the neutral stimulus doesn't help you predict, again, when the US is coming. It's completely predicted by the prior CS. So, there are biological constraints on learning. These are limits on learning. Some species come preloaded with certain behavior patterns, and some species are more sensitive to certain events than other species, whether those be CSs, USs, etc. There was an example on the wiki about blue sour water, or blue sour flavoring, or something like that, in color. And of course, some animals are going to be sensitive to color. Some animals are going to be sensitive to the flavor. There's a funny saying: "Never teach a pig to sing; it wastes your time and annoys the pig." Okay, last two things I want to talk about are discrimination and generalization. So what is discrimination? Let's say that I do a CS-US condition suppression preparation. There's an ongoing baseline of lever pressing, and I turn on a tone, or I turn on a red light and then present shock. And then at other times I turn on yellow light and give no shock. And then after a little while I turn on yellow light and give no shock. Then I turn on the red light and give shock. After a little while, I turn on the yellow light and give no shock. Yellow light, no shock. Red light, shock. Red light, shock. And then I test. What does the red light do? What does the yellow light do? And the results I get are the red light produces a conditioned response. The yellow light doesn't. That means if I turn on the red light, maybe lever pressing is suppressed to 0. The yellow light, there's no effect. It stays at its baseline rate. What this illustrates is what we call discrimination, or the way I define it is behaving differently in different situations. The rat discriminates red from yellow, or whatever animal this is. A rat probably couldn't discriminate red from yellow because it doesn't have the proper rods and cones in its eye to see wavelength. But nevertheless, if we were to do this experiment and this is the result, we would say that the rat is behaving differently in the presence of red than it is in the presence of yellow. It's behaving differently in different situations. Discrimination is defined experimentally. It's not what you want to happen, it's not what you hope to happen, it's not what you plan to happen. It is what the animal does. And a failure to discriminate is just that. Even you can arrange the greatest situations ever, but discrimination is behavior. It's behaving differently in different situations. On the opposite sort of side of the coin is a process we call generalization or generalization. We do the same procedure, but this in condition suppression, but in this case we just do red, red shock, red shock, red shock. And then in our test condition, we present red, yellow, orange, purple, blue, orange, red, yellow, orange, present all colors of the spectrum. And what sort of effects do we get? Well, what we see is that we see this condition response to the red is very great. As we present colors that are similar to red, we get pretty close to a peak condition response, but as we get farther away, we get less and less of the CR. This is a general principle of learning that we see almost universally. Is we see if we did training like this, we would expect a generalization gradient similar to this. I define generalization as behaving similarly in different situations. Okay, orange here is similar. Orange is different than red, but the behavior is similar. Purple is different than red, but the behavior around this is similar. Well, we're going to use the terms generalization and discrimination in operant conditioning and stimulus control as well. And there's many interesting features about discrimination and generalization. This is not a universal. Although we expect generalization to occur, we can affect it experimentally. That's one of the lessons we're going to learn. Okay, onwards and upwards. I'll see you in a little bit.